Table 6.6 lists some substances that are known to be either neurotransmitters or neuromodulators or both. This is actually a very short list because in reality there's really over 300 known neurotransmitters and neuromodulators, and the list is growing basically all the time. The bigger issue here is, as described in your textbook on page 166, is this whole issue of what's a neurotransmitter versus what's a neuromodulator. And that distinction isn't clear even now. I think one of the easiest ways to think of what's a neurotransmitter versus what's a neuromodulator is to look at the criteria that have been known for a long time that define what a neurotransmitter is. And there's three major criteria that have been used for many years in neuroscience to describe a substance or to classify it as a neurotransmitter. And the first one is that the substance must be present within the presynaptic neuron. Obviously, a chemical can't be secreted from a presynaptic neuron unless it's present there. And if it's present there, there's elaborate biochemical pathways in that neuron to produce it. And secondly, the substance that we're going to define as a neurotransmitter must be released in response to presynaptic depolarization, and that release must be calcium dependent. That's another essential criterion that neuroscientists have used for years to determine whether or not a substance is in fact a neurotransmitter. The third major criteria used is that specific receptors for the substance must be present on the postsynaptic cell. A neurotransmitter can't act on its target unless specific receptors for that neurotransmitter are present on the postsynaptic membrane. So these three criteria have to be met for something to be called a neurotransmitter. On the other hand, a neuromodulator it can be quite different. Neuromodulators don't even necessarily have to have receptors on the postsynaptic neuron. They can be thought more of as a substance that modulates the effect of the neurotransmitter. And the other criteria that I usually see in terms of what's a neurotransmitter and what's a neuromodulator is neurotransmitters, for the most part, are rapidly removed from the synapse by various mechanisms. Either an enzyme degrades them or a reuptake protein brings them back into the neuron that released them, or they're just simply washed away by flow of fluid from the synapse, from the synaptic cleft. But whatever the mechanism that's used, the neurotransmitter is typically only in the synapse for literally milliseconds, whereas neuromodulators are known to be present in the synapse for much longer periods of time because they're not being broken down or being reuptaken. So that's another way you can look at the difference between a neurotransmitter and a neuromodulator. Now, your textbook goes on to list some examples of a couple of very common neurotransmitters. They talk about acetylcholine. They talk a little bit about the catecholamines. And why don't we go to the next slide now, and I'll give you an example, a clinical example of for acetylcholine and its breakdown by acetylcholinesterase, and how this sometimes becomes an issue in medicine. Okay, what you're looking at here is actually figure 915 from your text. So you haven't seen this figure yet. You'll be seeing it when you get into chapter 9 and start talking about muscle. But I'm just using it here as an example of how neurotransmitters can be involved in either poisoning, or sometimes the same neurotransmitter can be involved in a medication that's actually useful for some type of disorder. And I've chosen to look at acetylcholine here because it's such a common neurotransmitter. As you'll learn more later when you get into chapter 9, acetylcholine, a neurotransmitter, is released into the neuromuscular junction with skeletal muscles, and it binds a type of acetylcholine receptor called the nicotinic receptor. When it does that, it creates an effect of causing skeletal muscle to contract. And again, you'll learn all about this in chapter 9. Like many neurotransmitters, or really all neurotransmitters by definition, after the acetylcholine is released into the neuromuscular junction, in this case is what it's called, it's rapidly destroyed by some type of enzyme. And in this case, the enzyme is called acetylcholinesterase. Well, as you can imagine, if you produced a compound that somehow blocked acetylcholinesterase or interfered with its ability to break down acetylcholine, what you'd end up with is huge amounts of acetylcholine in the neuromuscular junction or any other synapse where acetylcholine is released and broken down by acetylcholinesterase. Well, some poisons do this. For example, there's certain types of insecticides that are actually acetylcholinesterase inhibitors. So they do exactly what I just said. They inhibit acetylcholinesterase, and the result is that the insect finds itself swimming literally in a pool of acetylcholine in its body because the acetylcholine can't be broken down. And that becomes fatal for several reasons. And in humans, or in mammals in general, when you see acetylcholinesterase poisoning, for example, a child eats some type of insecticide, or even more commonly a dog eats it, the symptoms are what you would expect to see in somebody that has overactivation of their acetylcholine receptors, both muscarinic and nicotinic. They're salivating, there's tears coming out of their eyes, they're urinating, they have diarrhea, all the things that acetylcholine can do. Now, this can be rapidly fatal because they also can't breathe. They're swimming in their own secretions here in their lungs, and it will kill them, acetylcholinesterase poisoning, if you don't get them some type of acetylcholine receptor antagonist, such as atropine. That's the treatment for this type of thing. So acetylcholinesterase inhibitors can be very poisonous and can be extremely toxic to humans, and we see that in medicine. And again, the idea for a clinician is to simply be able to recognize it for what it is, either due to the history or for other reasons, and administration, in this case, of an acetylcholine receptor antagonist will be the thing that'll basically save this person. On the other hand, acetylcholinesterase inhibitors can also be useful if we want to have excessive amounts of acetylcholine in a synapse. And one example of that is in the treatment of Alzheimer's disease. Alzheimer's disease is a very complex disease, but certainly one component of it is the breakdown or degradation of neurons in the brain that use acetylcholine as a neurotransmitter. So to increase the effect of what acetylcholine releasing neurons are left in the brain of an Alzheimer's patient, they're given drugs that act as mild acetylcholinesterase inhibitors in the brain, which do exactly what I described previously, where they interfere with the ability of acetylcholinesterase in these neurons in the part of the brain that uses acetylcholine. And the result of that is that the acetylcholine that's released now in those parts of the brain is not broken down as rapidly, so it spends more time bound to postsynaptic acetylcholine receptors. And that seems to be beneficial, at least early on, in Alzheimer's disease patients. You may have heard of these drugs. The three classic ones that are approved in the United States are Tacrine, rivastigmine, and donepezil. So here you have an example of a neurotransmitter, acetylcholine, where pharmaceutically we're able to interfere with the enzyme that breaks it down, acetylcholinesterase, which results in lots of acetylcholine being around in synapses. In some cases, when you use a high dose or a certain type of acetylcholinesterase inhibitor, that makes it a poison. And in other cases, as I just noted, in the case of Alzheimer's disease, when you use certain types of acetylcholinesterase inhibitors at different doses, it can actually be a useful therapeutic modality. Okay, let's go on to the next slide. Okay, take a look at this figure 635 in your textbook. This just shows us the pathway for synthesis of the catecholamine neurotransmitters, and you should become familiar with this simply because the catecholamine neurotransmitters, there's tons of information known about them, and there's very, very many drugs that are used that act at the receptor for the catecholamine neurotransmitters, particularly norepinephrine and epinephrine. Those receptors are classified as beta and alpha adrenergic receptors. You may have heard of those. We have lots and lots of drugs in medicine that are either agonists or antagonists of beta and alpha adrenergic receptors. So having some knowledge of the synthesis of catecholamines is useful. The synthesis of these compounds is relatively straightforward. As you can see here, tyrosine, which is just an amino acid that's found in our diets, is actually transported across the blood-brain barrier and is taken up by certain neurons in the brain that make catecholamines and use them as a neurotransmitter. When tyrosine is taken up by these neurons, it's hydroxylated to L-DOPA by an enzyme called tyrosine hydroxylase, which is the rate-limiting step in this process. Now, once that occurs and the neuron is made L-DOPA, depending on what other enzymes that particular neuron has, that will dictate what the final neurotransmitter will be that it makes. You know, it may take L-DOPA and make it all the way and convert it all the way to epinephrine. If it's got the enzymes to do that, it may just convert it all the way to norepinephrine or just to dopamine. Again, it's dependent on which enzymes are present in that particular neuron that's utilizing tyrosine to make this neurotransmitter. Now, I'll give you a clinical example of how knowledge of this pathway has benefited human beings. Parkinson's disease, as probably most of you know, is a disease that's essentially a disease of too little dopamine, because a certain pathway in the brain that utilizes dopamine as a neurotransmitter has been destroyed by the processes that are involved in Parkinson's disease. So one of the therapies that's useful in the treatment of Parkinson's disease is dopamine replacement. And one of the ways that we can do that is to get dopamine into the brain of people that have Parkinson's disease. Well, we can't inject drugs directly into the brain. We give all drugs in the periphery, and then they have to cross into the brain. So we can't just give these individuals dopamine in the periphery because A, it doesn't cross into the brain well at all, doesn't cross the blood-brain barrier, and B, if you give dopamine in the periphery, even if it could cross the blood-brain barrier, a lot of it gets utilized in the periphery because there's dopamine receptors in the periphery, and it produces lots of unwanted side effects. So instead, Parkinson's patients are given L-DOPA, the dopamine precursor. And the idea is for L-DOPA, which is lipid-soluble and does readily cross into the blood-brain barrier, if we can give that to them, the hope is that that will cross into the brain and then be decarboxylated by DOPA decarboxylase to dopamine, where it can be utilized hopefully by the neurons that are dopamine deficient involved in Parkinson's disease. However, DOPA decarboxylase, the enzyme that converts L-DOPA to dopamine, is also present in the periphery, just like it is in the central nervous system. So when you give L-DOPA alone in the periphery, unfortunately, a lot of it gets decarboxylated to dopamine before it ever gets a chance to go into the brain and cross the blood-brain barrier. So typically, L-DOPA is given along with a peripheral DOPA decarboxylase inhibitor called carbidopa. So when you do that therapeutically to a Parkinson's disease patient, the carbidopa basically protects it in the periphery from being decarboxylated to dopamine, so it allows more of the L-DOPA to actually cross into the brain, and there it'll be converted by DOPA decarboxylase in the brain to dopamine, where it can be effectively utilized and help to alleviate the symptoms that you see in Parkinson's disease. So this catecholamine synthetic pathway is a good thing to know something about. So look it over and read the information that's in your textbook on pages 167 and 168, and just be familiar with the catecholamines, because we're going to be seeing them again in the future. All right, let's go to the next slide. Okay, this is figure 6.36 in your textbook, and this is showing what happens with release of the excitatory amino acid glutamate. Now, if you read this section of chapter 6, it's talking about amino acid neurotransmitters here. We've already discussed the fact that there's many different types of neurotransmitters that are known in the central nervous system. Some of them are catecholamines, as we've discussed. We've talked about other kinds of neurotransmitters. Some neurotransmitters are amino acids, and glutamate falls into that category. So glutamate is an amino acid neurotransmitter, but it's also an excitatory neurotransmitter, which means that when glutamate's released from a neuron and binds postsynaptic glutamate receptors, it causes that postsynaptic neuron to depolarize and fire. Now, what this figure is showing here is two major types of glutamate receptors that have been very well studied: AMPA receptors and NMDA receptors. I myself studied NMDA receptors for years for various reasons that I'll get to in a moment. So I'm familiar with these receptors just from my own personal experience with them. But the reason that this figure is in your textbook is it's showing one of the things that glutamate is able to do when it's released in certain parts of the brain, specifically the hippocampus, which we'll talk more about in chapter 8. It's able to invoke this process called long-term potentiation, or LTP. And LTP is very interesting to neuroscientists because it's known to be involved in memory and learning. And again, we'll talk more about that in chapter 8. But suffice to say, memory and learning is a very interesting phenomena in the medical profession, not only because we need to learn, and part of the process of learning involves memory, but also there's certain diseases that we get as we age, the most well-known one being Alzheimer's disease, where our ability to learn and remember is devastated. So learning how we learn is something that is highly studied in the area of neuroscience. So this is part of it, LTP. So you look at this phenomenon here, and it's showing glutamate being released from a presynaptic neuron and binding AMPA receptors and NMDA receptors on the postsynaptic neuron. The AMPA receptor is a classic ionotropic receptor, meaning that when glutamate binds to it, it opens an ion channel that the AMPA receptor is associated with. In this case, the ion channel is a sodium channel. So glutamate's release binds to the AMPA receptor. It opens this binding causes the ion channel that the AMPA receptor is associated with to open, which, as I said, is a sodium channel in this case. Sodium flows in and begins to depolarize that postsynaptic neuron. Well, NMDA receptors are not classic ionotropic receptors. So NMDA receptors are also associated with a channel, but in this case, with NMDA receptors, it's a calcium channel. So in other words, when NMDA receptors open up the channel that they're associated with, it's a calcium channel, and calcium flows into the neuron. But interestingly, in LTP, NMDA receptors there has to be significant depolarization of the neuron before NMDA receptors can actually open this ion channel, this calcium channel. And it's because a magnesium ion sits in this NMDA receptor associated channel, and it blocks calcium entry. So what happens here, and this is, as you can see, is very complicated, but in a nutshell, what happens when this phenomena occurs is that glutamate's released, it binds to AMPA receptors. Those receptors quickly open sodium channels. Sodium flows into the postsynaptic neuron and begins to depolarize it. Once enough depolarization has occurred, i.e., once the inside of that postsynaptic neuron has become more positive, then the binding of the magnesium ion is kicked out of the NMDA channel, and now the binding of glutamate to the NMDA receptors can open the NMDA receptor associated channel, and calcium flows in. So both of these phenomena are required in the process of LTP. And again, glutamate acts like other excitatory neurotransmitters in certain parts of the central nervous system. It's only in certain areas of the brain, as I said, particularly the hippocampus, where it not only just causes depolarization of postsynaptic neurons, but it causes this phenomena of LTP. And that phenomena, again, is what's being described here, where AMPA receptors open up sodium channels, the neuron's depolarized, and once it's depolarized enough, NMDA receptor associated calcium channels open up and calcium flows in. And then there's, of course, subsequent events that occur in that postsynaptic neuron that are involved in learning and memory that we'll discuss a little more in chapter 8. But this phenomena, it's shown here to describe LTP, and you can read about it on page 169 in your textbook. I don't expect you to memorize this process of LTP. I just want you to know the concept of it, because it's very, very difficult concept. Whole books, entire manuscripts are written just on LTP. Another thing that's interesting about glutamate and its receptors, in particular the NMDA receptor, I studied it in the context of neuron toxicity. The NMDA receptor, it's well known now, is involved in neuronal cell death in certain situations, particularly ischemic brain disease, such as occurs after a stroke. When a stroke occurs, basically parts of your brain are not getting blood anymore, and therefore they're not getting oxygen and nutrients, and that part of the brain starts to die. And when that part of the brain starts to die, the neurons that contain glutamate, when they die, they rupture and they release all of their glutamate into the surrounding interstitial fluid, cerebrospinal fluid, and surrounding synapses. This causes overstimulation of NMDA receptors in that area because of all this glutamate that's being dumped from dead neurons. That overstimulation of NMDA receptors then causes lots of calcium to flow into these surrounding neurons, too much calcium, and results in their death. So the bottom line is you have way too much glutamate around when neurons are dying, overstimulating NMDA receptors and killing neurons. So that's why after you have a stroke, the damage to your brain doesn't occur immediately. It typically occurs over 2 or 3 days, because this is progressive. As neurons die, dump glutamate. More neurons die, they dump glutamate. So one of the treatments now for a stroke is to give people an NMDA receptor antagonist. And NMDA receptor activation isn't the only thing involved in neuronal cell death, but it certainly is a player. And that's what I was studying for a number of years. We were creating compounds that would act as NMDA receptor antagonists that could be used in people who had a stroke. Okay, that's enough of that. So let's move on now to the next slide. Okay, this starts the last section of Chapter 6, Section D, which has to do with the gross structure of the nervous system. This will come in useful for us when we get into Chapters 7 and 8, where there's a lot of, in the case of Chapter 7, sensory physiology, and in Chapter 8, issues with the higher brain in terms of learning and memory and consciousness. You should look at this beginning of this chapter and memorize some of these definitions. For example, if you look at this figure here in this particular slide, it's figure 637 from chapter 6 in your text, and it just shows the major breakdown of the nervous system, where it's subdivided into the central nervous system, which is the brain and the spinal cord, and the peripheral nervous system, which is basically all parts of the nervous system that are outside of the brain and the spinal cord. And as you can see here on the right hand side of this figure, the peripheral nervous system is subdivided into an afferent and efferent division. The afferent division, of course, bringing essentially information into the central nervous system, and the efferent division bringing information out of the central nervous system to the peripheral nervous system. So I think it goes without saying that the peripheral nervous system and the central nervous system communicate with each other, and we'll be looking at how that's done in this section. So some of this information here that you should review, because it's terminology that's used a lot in this section and will be used in subsequent chapters. For example, recall that a long extension from a single neuron is called an axon or a nerve fiber, and we use the term nerve per se to refer to a group or a cluster of axons that are traveling together to and from the peripheral nervous system. So in other words, there aren't any things that we call nerves in the central nervous system. Nerves are cables or clusters of single neurons that are bundled together that we find in the peripheral nervous system. There's a lot of them. We'll talk about some of them. When nerves are clustered together in the central nervous system, we call it a pathway or a track. And we'll be talking about some of those, especially in Chapter 8 when we talk about consciousness and higher parts of the brain. And you should just review this because we talk about tracks, we talk about nerves, we talk about commissures. So just go over that stuff at the beginning of Section D so that you're familiar with it. All right, after you've digested this a little bit, let's move on to the next slide. Okay, this is figure 638 at the beginning of Section D of Chapter 6, and this is like the next slide as well. It's basically an anatomy slide, and this isn't an anatomy course, as you know. This is a physiology course, so you don't have to memorize any of this anatomy of the brain here that they're showing, but you should review it because in subsequent chapters, especially 7 and 8 in particular, we talk about specific physiology, like learning and memory, for example, in Chapter 8, that's going on in certain parts of the brain. And so you should become somewhat familiar with the gross anatomy, but again, in terms of exams, I won't be asking you to regurgitate specific anatomy. But as you look at this picture, just become familiar with the fact that the brain is essentially divided into 4 regions: the cerebrum, the diencephalon, the brain stem, and the cerebellum. And it's particularly the cerebrum that we'll be studying in this course, and that's subdivided into a right and left cerebral hemisphere, and we'll be looking at that in the next slide and breaking that down a little bit. But as you look at this thing, be aware of the fact that as the spinal cord enters into the skull, at the base of the skull, it first forms the brain stem, which, as you can see here, is the midbrain, the pons, and the medulla, with the cerebellum sitting posterior to that area. These areas of the brain are predominantly function with regards to unconscious things that are going on in your body, unconscious functions that you don't consciously control, heart rate, etc. If this part of the brain is damaged, this brain stem, generally it's lethal, because the rest, so many parts of your body and organ systems that are innervated by the brain, this part of the brain stem, controls those autonomic functions. Above that, as you see in this picture, sits the forebrain, as we said, is the cerebrum and the diencephalon, and that's where the higher learning, or humans as the advanced species that we are, a lot of our intelligence comes from our well-developed cerebrum, making us able to learn things that other animals cannot learn. So again, look this over and just become familiar with the 4 main regions of the brain, the 4 subdivisions. And now we'll go and look at the next slide and break down a little bit. We'll look at the coronal section, showing the forebrain and basically the gray matter and the white matter. Okay, figure 639 here from your text is simply showing a frontal section of the forebrain. As you look at this thing, keep in mind that this is the forebrain, and it is composed of the cerebrum and the diencephalon. And the diencephalon is basically the thalamus and the hypothalamus that's shown here in this figure, and the rest of it is the cerebrum. And the cerebrum is basically where a lot of the action is in the brain, as we're going to learn soon, in terms of not only learning and thinking, but all of our senses as well. The cerebrum, as you can see here, is divided into a left and right cerebral hemisphere, and the cerebral hemisphere itself consists of a cortex, which is an outer shell, gray matter. It's always drawn as gray in pictures because it looks gray, and the reason it looks gray is because cell bodies of neurons are clustered in this area, and they give a gray-like appearance. And then underneath this gray matter is what's called white matter, and white matter is simply tracts of nerve fibers that are myelinated and give it that white appearance. And the cortex of each cerebral hemisphere here too is divided into 4 lobes, and you can actually see those lobes better in the previous figure 638. But there's the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobes. And the cortex is very thin. Your book says it's only 3 millimeters thick, but other texts say 4 or 5 millimeters thick. But it's highly folded, so it has a lot more surface area, the cerebral cortex, that it would not otherwise have if it wasn't folded. So there's quite a bit of surface area here, and of course the cerebral cortex, as I said, does many functions that we're going to be looking at later. In this gray matter here in the cerebral cortex, you can see in this figure that it shows that the cells are organized in 6 layers basically. And these neurons that compose these cells are of 2 basic types: pyramidal cells and non-pyramidal cells. And you can see these layers again drawn in the upper right corner here that compose this gray matter. Again, as I said earlier, the cell bodies of these neurons are what give the outer cortex here its gray-like appearance. And these pyramidal cells, as you'll see later, play a major role in several functions, but in particular in learning and memory. So again, a lot of anatomy here. You just need to become vaguely familiar with it. You can use these figures as a reference as we go through other chapters and we start talking about this sense or that sense, vision, hearing, taste, etc. And then they begin to refer to various parts of the cerebral cortex, such as the frontal lobe, or the parietal lobe, the occipital lobe. And then you can come back and you can look at these figures if you've forgotten the anatomy, to just kind of remember which part of the cortex is involved in what. The reason this is interesting, by the way, from a clinical perspective, if any of you ever go to med school and you take a neuroanatomy course, you'll see that it's the reason that we know which parts of the brain are involved in which senses and which type of learning, etc., is because when people damage them for whatever reason, they lose the ability to do certain things, whether it's to smell or taste or speak, etc., or think. And over the decades, as we've become better and better at imaging the brain, when people do injure their brain, we can see which parts of the brain are injured, and then that is correlated to what subsequently is wrong with the person. Like, as I said, inability to move their muscles, inability to speak, etc., etc. So that's how the functions of the cerebral cortex were initially determined, by correlating loss of some function with specific damage of some part of the cerebral cortex. All right, let's go to the next slide. Okay, this is figure 640 from your book, showing the limbic system and the functions of the limbic system. The limbic system is drawn here in purple. You see the orange and dark purple structure here, the thalamus and the hypothalamus, thus named because it sits underneath the thalamus, which are composed of diencephalon. The limbic system is another extremely complex system. There's no part of the brain that I've ever seen that isn't extremely complex. The limbic system, though, has been referred to for many years as the emotional brain, the emotional part of our brain. It's highly involved in emotion and learning, appetite, which is a visceral function, sex, or any type of rewarding behavior that you will do again because it feels good. Drug addiction being another example, is mediated through the limbic system, and also endocrine integration, as you'll also see later. So the limbic system is very complex, and it involves these structures that you see here: the hippocampus, the ones that aren't drawn here are the amygdala and the parts of the amygdala that are important. The amygdala is part of the limbic system, and it's an extremely important component of the emotion fear, and to some extent anger as well. That's not drawn on here. The hippocampus, which is drawn on here, as we'll see later, especially in chapter 8, is extremely important in the ability to form memories, as I've already alluded to when we talked about long-term potentiation a few slides ago. So again, in this whole section, section D, we do a lot of anatomy, and these slides will mostly be used as references, so that when we get into chapter 7 and 8, and we start talking about the olfactory system in terms of smell, etc., etc., you can come back and look at this stuff and see, remind yourself what exactly structures compose the limbic system, and which structure in particular we're talking about when we get into some of that physiology. At this point, let's just go to the next slide. Okay, this is figure 641 from your text. This is some basic anatomy of the spinal cord and the surrounding vertebrae. Up to this point, we've been talking about anatomy, neuroanatomy here in section D of this chapter, but it's been the anatomy of the brain. So this is the first slide that shows some anatomy of the spinal cord, which is, of course, the other part of the central nervous system. You look at this thing, you can see that the spinal cord itself you can't tell from this figure because it's not drawn to scale, but the spinal cord itself is only about the size of your little finger. The vertebrae, the surrounding vertebral column that follows it in the periphery, is there for protection and support. But as you look at this thing, you can see the spinal cord per se has gray matter and white matter, just like the brain does. It is because it is part of the central nervous system. The gray matter in this case is composed predominantly of interneurons, and it also contains the cell bodies and dendrites of efferent neurons, and it contains the axons of entering afferent neurons. And there's several types of glial cells that are present here in this gray matter as well. You can see it looks like a butterfly, and anatomically, anatomists refer to the dorsal horn is the part of the butterfly that projects towards your back or to the dorsal part of your body, and the ventral horn is the part of this gray matter that looks like a butterfly that projects towards your anterior part of your body or the ventral part of your body. The white matter surrounding the gray matter, as it does in the brain, are myelinated neurons. They run in tracks up and down the spinal cord. Some of these myelinated tracks run up to the brain. They ascend up to the brain, and some of them descend from the brain down to the spinal cord, bringing information from the brain to the spinal cord. Now, if you look closely at this particular figure, you can also see, interestingly, that the dorsal root and the dorsal root ganglions bring information from the periphery into the spinal cord. So you have a dorsal root bringing information in on the dorsal side of the spinal cord, and the ganglion simply contains the cell bodies of these afferent neurons that are bringing information into the spinal cord, while the ventral roots here contain information that's leaving the spinal cord and going towards the periphery. And you can also see here that the dorsal roots and the ventral roots fuse very short distance from the spinal cord to form a spinal nerve on each side of the spinal cord. So this particular phenomena, this issue of dorsal and ventral roots and dorsal roots containing neurons bringing information into the spinal cord, and ventral roots containing information coming from the spinal cord to the periphery, and then them subsequently forming spinal nerves, becomes a little more relevant shortly when we start talking about the autonomic nervous system. At this point, though, let's leave this slide and go to the next slide. Okay, now we're going to start looking at some of the structure of the peripheral nervous system, specifically here in table 6.8, we're looking at the cranial nerves. Now remember I said earlier that when we talk about nerves per se, those things are only located in the periphery. And a nerve, as we discussed, is basically a group of thousands and thousands of nerve axons that are bundled together in something that looks like a little cable. And inside these nerves, what we call nerves in the periphery, some of these nerves may be afferent or sensory nerves. Some of these nerves may be motor nerves or efferent nerves. Some nerves are just one or the other. Other nerves have both mixed afferent and efferent effects, as you'll see. Now there's 43 pairs of nerves in the peripheral nervous system, and again we're looking at the 12 pairs of cranial nerves here in table 6.8. The cranial nerves are interesting in that they actually come out of the brain stem. They don't exit from the spinal cord per se. They exit from the brain stem. And if you've forgotten what the brain stem is, look back at the beginning of section D here at figure 6.3.8 to remind yourself. These cranial nerves can have either afferent or sensory, carry afferent or sensory information, or efferent or motor information, or both. And if you look at these, you'll see that the ones that are, there's 3 of them that are predominantly sensory or afferent in their function, and that's cranial nerve 1, the olfactory nerve, and cranial nerve 2, the optic nerve, and cranial nerve 8, the vestibulocochlear nerve, which is involved with hearing and balance. There's 4 of these 12 that are primarily motor nerves or efferent nerves, and that's cranial nerve 4, the trochlear nerve, cranial nerve 6, the abducens, cranial nerve 11, the accessory nerve, and cranial nerve 12, the hypoglossal nerve. And finally, there's 5 of these cranial nerves that are primarily mixed, what are called mixed nerves, that have both sensory function or afferent function and motor function or efferent function. And these are cranial nerve 3, the oculomotor, cranial nerve 5, the trigeminal, cranial nerve 7, facial nerve, cranial nerve 9, the glossopharyngeal nerve, and cranial nerve 10, the vagus nerve. So I don't expect you to memorize all of these nerves and all of these functions of these nerves, but I just want you to recognize the fact, you need to understand that there are in fact 43 pairs of peripheral nerves. 12 of them are cranial nerves, and as we'll see in a minute, we'll look at the 31 pairs of spinal nerves. If you are going to go to medical school, of course, you will have to memorize all these these cranial nerves, and there's different mnemonics to do that. One that I like, if you are interested in memorizing them now, is once one takes the anatomy final, very good vacations are heavenly, is a mnemonic that I've used in the past to memorize the cranial nerves. All right, at this point, let's leave this table 6.8 on the cranial nerves, and now let's look at the spinal nerves, the 31 pairs of spinal nerves.